Welcome, everyone, to the new and improved virtual epidemiology brought to you by COVID 19, social isolation, and flattening the curve. What does all that mean? Well, we'll get there because we're talking about infectious disease epidemiology. The world right now is going through a bit of a crash course in epidemiology, whether they want to or not. They've got R naughts, reproductive numbers to the left. They've got mortality rates to the right. And everyone's talking about herd immunity and flattening that curve. So, what does that all mean? Well, we're going to get to that today when we talk about some basic principles of infectious disease epidemiology. We'll follow this lecture up with a more specific discussion of pandemics and specifically pandemics that come from animals. But right now, let's review a few concepts and introduce a few others. We've learned about the epidemiology triangle, but I think it's worth revisiting now that we have experienced this real life case study that is COVID 19. Talking about the chain of infection. So, how do infectious agents circulate around in people? Uh, and then finally, we're going to end with a discussion on herd immunity, which has to do with that whole curve flattening thing that we're all so aware of now. So, simply put, an infectious disease is a disease or infection that is caused by the invasion of a host by some agent. So, the agent could be a virus, a bacteria, or a parasite that essentially hijacks the host and causes harm. For example, if it's a virus, it kind of hijacks the cellular mechanisms to produce to get the body to produce more viruses. But the point is, it is some foreign agent that harms the host, and that this harm is what causes disease. And these agents can be passed or transmitted to other people. So, i.e., they are infectious diseases, as opposed to non communicable or non infectious diseases that cannot be passed between people. So we got our guy Jon Snow. He's over here sheltering in place with us. He's providing us some much needed sage advice during all this kind of madness. 6 feet away, Jon Snow. You know the rules. The epidemiology triangle. We've talked about this before, but it's worth revisiting. The epidemiology triangle is a very basic framework for understanding why disease exists in certain locations. It consists of three components: the agent, i.e., the infectious foreign pathogen, such as a virus, a bacteria, parasite. It consists of the susceptible human host. As you will recall, this is my first baby photo, and I was quite unhappy because I couldn't quite grasp at that time what the utility or what the purpose of the epidemiology triangle was. But I'm feeling much better now because I think I know what this is all about. Okay. So we can't have disease without people. Okay. You can have a location where viruses and bacteria are floating around everywhere, but if there's no people there, you're not going to have disease. Okay. And then finally, let me just erase this. Finally, we need an environment that allows the host and the agent to come together. Okay. So if it's a disease like malaria that requires a little help from a mosquito to transmit to people, you're talking about an environment that requires a certain amount of rainfall, a certain temperature, even certain types of trees and plants, and even animals. So this is obviously specific to each unique disease, and we can use this triangle to really understand a disease like, let's say, COVID 19, that is occurring in certain clusters throughout the world and throughout the country. If there are a lot of cases in a certain location, it means that this triangle is being completed at a higher rate in those areas. So we might want to ask, What is it about the environment? What makes the environment maybe different than other places that don't have the disease as much? So, this is a very useful tool for helping us understand how and why disease spreads. And so, what do we do with all this information? The idea is we need to know something about these factors so that we can ultimately break the transmission cycle. Okay. If we know, for example, that a particular disease requires standing water, so like pools and puddles of water, for example, the mosquito to breed in and pass a disease to us, we can do a little intervention there by maybe draining standing pools of water and eliminating that connection. We can go spray for insects. We can use insecticide and spray to try to kill mosquito populations. That gets rid of the vector. If we want to try to block the pathogen or the agent from infecting the host, we can add a bed net. So that's a physical barrier between the person and the mosquito vector with the infectious agent. Okay. So this helps us understand not only how and why the disease is spreading, but also like what to do about it. How do we design an intervention to break apart this chain? Because if any one of these linkages is broken, we don't get disease. Okay. So we're talking about infectious diseases specifically now. Let's talk about specifically this agent. How does this um, foreign pathogen get to us, the person? So Jon Snow wants to know, what is all this chain of infection business I've been hearing about? Basically, Jon Snow, the chain of infection describes the ways or the mechanisms in which those agents infect the host. So the chain of infection, similar to the epi triangle, is basically a series of linkages starting with the infectious agent itself. Think about virus, fungi, bacteria, etc. And follow the chain through to the susceptible host, which ends in disease. Okay. And it's up to us, public health warriors that we are, to design interventions to break the chain. Okay. For example, if we know that a disease is spread through close contact, maybe through shaking hands, uh, maybe we want to break the chain by improving hand hygiene or cleanliness measures. So we're going to walk through a few of these right now. So we talked about the infectious agent a little bit, but we're gonna get into some of the categories that define what an infectious agent is. We're going to talk about, okay, where do all these things come from? The disease reservoir, the portal of exit. How do they get out of the reservoir and how do they make it out into the big scary world? Once they're out, how do they spread? What is their mode of transmission? Are they directly spread from person to person, or do they require a few in between steps? What is their portal of entry? How do they make it into the susceptible human host? And then what makes a host susceptible? So let's start with a little discussion about the different ways that epidemiologists basically like to group infectious agents. And we're going to talk about a few broad categories. So, one of the common forms of infectious agents are bacteria. Bacteria are unicellular prokaryotes, and for you bioenthusiasts out there, that means that they have no membrane bound nucleus, and they basically reproduce by growing and dividing through a process called binary fission. We can broadly categorize bacterial agents in one of two ways. They can either be gram positive or gram negative, meaning when a special stain is applied, we can look and see different colors under a microscope. Basically, what this allows us to do is just narrow down the list of possible culprits, especially like if we're identifying or investigating a disease outbreak. We can look for a bacteria, and if we can tell that it's gram positive, for example, Well, we can eliminate all the gram-negative suspects right away. It's just a way of us kind of narrowing down the list of culprits. And in addition to categorizing bacteria by whether they stain gram-positive or stain gram-negative, we can further identify them by different shapes. So some of them are kind of like rod shapes. Some look like little clusters of grapes. Some are more kind of spiral with little kind of uh, arms coming out of them. There are a few different ways we can kind of classify bacteria. We're not going to get too much into the weeds here, but here's an example of what you might see when you're reading like an epidemiological paper or something. So if we're talking about community-acquired pneumonia, for example, it is a pneumonococcus, meaning it has this kind of little cluster grape shape, and it is a gram-positive bacteria. So we know that if it's applied to this certain stain, it will stain gram-positive. Okay, so this is a way of classifying this agent. The other main category of infectious agents are viruses. Viruses are everywhere you don't want to be. Viruses are found everywhere and they are found in everything. Viruses infect all living organisms and they have been with us since the beginning of time. A virus is an interesting agent because there's quite a bit of debate as to whether it's actually even alive. Uh, the reason for that debate is it doesn't technically reproduce or like divide on its own like a bacteria does. It needs to find a host cell and it basically just invades it and hijacks it and turns it into a little virus factory. So essentially, viruses are pretty simple. They're just little clusters of nucleic acid surrounded by a protein coat. Okay. And if you think of all these little spikes on the outskirts of the virus, think about these spikes as kind of like a key. And your cell in your body is a certain kind of a lock. And for a virus to invade your cell, that key needs to fit into the correct sized lock. So that's why not all viruses infect everyone or everything equally. And for example, why some viruses can only infect cells in the respiratory tract, because those cells, the lock and key shape is a certain way. We can talk about viruses in one of two ways. There are DNA viruses, double or single stranded DNA, and there are RNA viruses. RNA viruses, you'll sometimes see the word retrovirus. Herpes virus is an example of DNA, and HIV is an example of an RNA virus. So we're not going to get too much into that right now, but it's just another way of further classifying the infectious diseases that we're talking about. And here's a few Snapple facts for you, and I know you missed those. 1 milliliter of seawater contains 1 million virus particles. Let's just let that sink in for a second. So next time you're looking to go out to the ocean, um, you know, maybe think about this. And like I said, viruses are not technically alive. They don't have cells, and they can't technically turn food into energy, which is like a basic definition that we have for what is life, right? Um, And because of this, there's this ongoing debate. Doesn't matter. It's basically kind of like a nerd fight between those who think they are alive and who aren't. We don't care right now. We just know that there are some things that can get us sick, and they're called viruses. Fungi, the fungus among us. The third classification of infectious agents. Fungi, for example, ringworm over here, that's you can see why it gets its name there, are eukaryotic, meaning that their nucleus is enclosed with a membrane, and they have cell walls, rigid cell walls. They produce by forming spores, and they are a little more complicated in that they are multicellular. So we're starting to get a little more complicated here. They play an important role in the environment. We need fungi around because they are natural decomposers. They help break down organic material and help the circle of life. Protozoa, the parasites. So this is the year of parasites. Parasite won the Academy Award for Best Picture. If you haven't seen it, it's kind of weird, but I liked it. Protozoa are unicellular, heterotrophic, meaning they basically have to eat other plants and animals for energy and nutrients. That's why they're called parasites. They don't have cell walls, so they can kind of squirm around and they have rapid, flexible movement. And they are typically found in contaminated food and water, but they can also be transmitted through the bite of an insect. Malaria is a good example of a protozoa. Plasmodium are parasites, and there are different types: Plasmodium falciparum, Plasmodium vivax. The list goes on. Helminths. These are even more complex. They are tiny little invertebrate animals. Some helminths can be parasitic. They are multicellular animals, so the medications used to treat helminth infections can produce pretty negative side effects because these drugs can be toxic to humans. They have more complex reproductive cycles. This is an example of the reproductive cycle of schistosomiasis, which is a fun word to say, and I encourage you to try to say it 5 times fast. Schistosomiasis requires a more complex life cycle that involves snails. We'll get into it. Don't worry. Okay, so we talked about the different ways that we can categorize infectious agents. And not all infectious agents are basically created equal in the eyes of an infectious disease epidemiologist. How serious should we be taking this virus or that bacteria? Basically, how badly is it going to get us sick? How quickly is it going to spread? Is it going to kill us? Infectivity of an infectious agent is basically how likely the infectious agent is to spread. How likely is the agent going to infect a host that has been exposed? This can be measured as the basic reproductive number, and I'm going to come back to that in a second. Pathogenicity. How able is the agent to cause disease? This means if a certain infectious agent is more likely to cause harm to the host, it is said to be more pathogenic. We can measure pathogenicity as the ratio of apparent to inapparent infection ratio. So what does that mean? An infectious agent with more pathogenicity is more likely to get you sick, visibly sick, symptoms, coughing, sneezing, headaches, don't feel good. Okay. The more people that are exhibiting symptoms compared to the people who are maybe infected but not exhibiting symptoms, that is what that ratio means. Virulence. How likely is the agent to cause severe disease? This is measured as the case fatality ratio. So, with the COVID-19 pandemic, a lot of attention has been paid to what is that case fatality ratio? What is that case fatality rate? What is it doing? If a case fatality rate is 3%, it means that 3 out of every 100 people who are infected with the disease will die from the disease. Obviously, as that number goes up, the infectious agent is said to be more virulent. And this is, again, a measure of disease severity. So that's a little bit about these little agents, these little pathogens that are causing all this trouble. So where do they come from? And we can talk about and classify infectious agents in terms of 4 broad categories. Some infectious agents actually come from humans and spread from human to human from there. Some looking at you, COVID-19, come from animals, and they are said to be zoonotic. Others come from soil. They are dirty. Uh, Anthrax comes to mind. And then some come from water. So our boy Jon Snow knows all about that when he investigated the cholera outbreak in London. So humans, animals, dirt, and water. These infectious agents don't magically appear. They come from somewhere. Here's where. So Jon Snow wants to know, how contagious are these different diseases? They don't all spread equally, do they? Well, no, Jon Snow, and I shame on you for not remembering. The R naught is the basic reproductive number for the infectious disease. This is a number that has been circulating around the media if you've been paying attention to the pandemic, which I hope you are, as an informed citizen and epidemiology enthusiast that you are. The R naught, or basic reproductive number, is basically how fast a disease spreads. It is the, quote, number of secondary cases produced by a single infection. And here's the caveat: in a completely susceptible population. Well, what does that mean? If I get 2 people sick, I have an R naught of 2. Then each of those people get 2 people sick, and so on. If I have an R naught of 18, if I walk into a coffee shop, I'm basically going to pass the disease to every single person in there. And then each of those people are going to pass it to 18. And the curve over time goes up rapidly. An R naught that is less than 1 means that, on average, one infected person will produce less than 1 infection. When this happens, the disease dies. The outbreak ends, and we can resume our regularly scheduled programming. If the R naught is greater than 1, such as what we're experiencing now, every individual who's infected produces, on average, more than 1 new infected individual. The disease invades a susceptible population. So if everyone is a susceptible host, if everyone can theoretically get the disease, for example, with COVID-19, you have a completely susceptible population. And that is even more apparent when you have a new infectious disease without any vaccine or prior human exposure. Everyone is susceptible. Therefore, when we have an R naught of, let's say, 2, for every person who gets COVID-19, they will on average pass it to 2 more. We'll talk more about that in a second. So the magic number we want to hit is 1 or less than 1. Okay? As long as that R naught is greater than 1, disease will continue to spread in a population. Different things impact or affect the R naught. Why does the R naught go up or down? Why does it change over time? Why do some diseases have higher R naughts than others? Depends on a number of different things. We can compare diseases by their R naught. Hepatitis C, Ebola, for example, these have R naughts around 2. HIV, SARS, and you all should know SARS at this point. SARS is another virus in the coronavirus family. We are experiencing COVID-19, which is the 7th known human coronavirus. SARS happened 10 years ago or so, and it had a reproductive number of around 4. Measles is the most contagious disease we know about. It has an R naught of 18. One person with measles, on average, will pass it to 18 other people. That's scary. So why do these different pathogens or different diseases have different R naughts? Why is Ebola a 2 and measles is an 18? Well, part of it has to do with how the agent spreads. Ebola is spread from close contact with bodily fluids, like blood. Measles is spread through the air. So it's easier to get the infected agent into basically a fine mist spit cloud, and it travels around and it can get a wider group of people sick. So a disease like hepatitis C and Ebola, those are a little more close contact, so they're a little slower to spread. So if we say that the R naught, for example, in measles is 15, then we would expect it to spread very, very quickly because each new case would produce around 15 secondary cases. This is why vaccinations are so critical, because if just a few people don't get vaccinated against a disease like measles, it really doesn't take much for the disease to just take off because it is so contagious. So, the way that a pathogen spreads will affect its R naught. Diseases that are spread either through contact, directly or indirectly, um, those that are spread through vectors like mosquitoes or ticks, those that are airborne, and then some spread through food and water. So when the pathogen leaves the reservoir, whether it's a person or a pond or a bat, it gets to the susceptible host through one of these modes of transmission. Okay, and we're going to talk about a few here. Direct contact mode of transmission requires direct, close physical contact between an infected person and a susceptible person. It includes skin to skin contact, kissing, sexual contact, or close contact with body lesions. HIV is an example of a disease that requires direct contact. It's another reason why HIV doesn't have a very particularly high R naught, because it's a little slower and harder to pass the disease compared to a disease that is spread through the air. Droplets. Droplet spread is a disease transmitted by large infected droplets. So think about coughing, sneezing, these ways that humans can produce these little spit clouds, essentially. Think of this as like the spray zone. Think of this as why, for example, with COVID-19, you are instructed to stay 6 feet apart from other people, because 6 feet on average is a human splash zone. That's how far a sneeze or cough particle or droplet potentially could spread. So these are diseases like pertussis, tuberculosis, COVID 19, and they are a little different from some of these other modes of transmission, like indirect contact. Indirect contact transmission occurs without direct human to human contact. It typically involves the transfer of pathogens from a reservoir to a susceptible host via a vector, whether it's an insect or a doorknob. A doorknob, what are you talking about? I'm talking about a fomite. Fomites are inanimate objects that become contaminated with a pathogen. And let's use the example of a doorknob. If I am infected with a respiratory disease and I cough into my hand, I have some virus on my hand. And if I go to open the door, that virus gets transferred to the door handle. If the next person behind me is healthy and perfectly susceptible, they touch that door handle, and then what do people do? They scratch their face, they touch their face, they pick their nose, they pick their teeth, they infect themselves. And that process occurs not because the infected person directly transmits it to the susceptible person, it's transmitted via the door handle. In that case, the door handle becomes a fomite. Fomites can be anything that allows the transfer of pathogens between infected people and susceptible people. Fomites are why I recoil in terror when I see this. I don't like that. The reason I don't like that is because of fomites. Fomites are a big issue right now with the pandemic because that is one of the ways that it's spread. This is why everyone is going around saying, hey, wash your hands. Did you wash your hands? How many times have you washed your hands? This is in direct response to the fact that COVID-19 can be spread through fomites, meaning if I'm infected with the disease and I sneeze into my hand and I go to open the door, that virus could theoretically stay infectious on that door handle, waiting for the next person to come. If you were to touch that door handle and it contained the virus on it, the only real way to protect yourself in that case is to wash your hands with soap and water to prevent the virus from infecting you. Why soap and water and why not, I don't know, a gallon of bleach or nail polish remover or napalm? Well, viruses are pretty picky little things. And essentially, what a virus is just some genetic material held together by some oil. And what cuts through grease and oil? Soap. So when you expose a virus to soap, it basically dissolves. So airborne droplet nuclei are a little bit different than these big droplets that we just talked about. For example, from sneezing or coughing, airborne droplet nuclei are finer and get suspended in the air for longer. And they can actually stay kind of floating in the air for long periods of time. Some of these can even get sucked up into air ducts and travel around from room to room. So if you produced airborne droplet nuclei in one room, you could potentially infect someone on another floor or a room down the hall. Uh, Measles is spread this way. This is why measles has such a high R naught, because measles can stay suspended in the air for longer periods of time. If I have measles and I produce a cloud of airborne droplet nuclei through a sneeze, that measles pathogen can stay floating in the air for several hours after I've left the room. If you go in to that room that I was in several hours earlier, you could theoretically become infected. Terrifying. Some pathogens need a little help. They need a little help from an insect like a mosquito or a tick or some other kind of go between. Vector borne diseases are what we're talking about here. Malaria is a vector borne disease. Lyme disease is a vector borne disease. Lyme disease is transmitted from an animal host or animal reservoir to a susceptible human through the bite of a tick. So, malaria, Lyme disease, plague, these are all examples of vector borne diseases. COVID 19 is not a vector borne disease. So, don't worry about that. Okay, how else do we talk about infectious diseases? Well, what do they do to us? How do they get us sick? We can broadly classify infectious diseases by also their clinical classification. How do they get us sick? Well, they can give us diarrhea. They can produce respiratory disease, such as COVID 19. They can cause cutaneous or soft tissue infection, such as leishmaniasis. They can cause central nervous system disease, such as so called mad cow disease. Septicemic disease causes infection. Think about sepsis. And then the so called fever of undetermined origin, kind of that miscellaneous category. So, Jon Snow wants to know a little bit more about herd immunity. We kind of teased at this earlier, and he's just chomping at the bit because Jon Snow knows that herd immunity is the key to stopping an infectious disease outbreak. Herd immunity is what public health folks are all about. Herd immunity is why we have flu shot campaigns. Herd immunity is when we have enough people in a community who are immunized against a disease that the entire community is safe. So let's imagine that I am infected with, let's say, a new strain of flu, and I walk into a classroom. Um, Remember when we could do that? I miss those days. I'm infected with influenza. I walk into a classroom, and let's say that none of you guys got your flu shots. Everyone is potentially susceptible. And if I sneeze, there's nothing really stopping that virus that I have from infecting maybe the people in the front row. And then maybe they infect the people behind them. The disease can spread very, very quickly. Let's think about another scenario, which you guys are all good herd immunity public health practitioners. And let's say that everyone got the flu shot. I walk in and I sneeze, that virus has nowhere to go. Everyone is immune. Let's say that um, let's say that someone sitting in the back of the room maybe couldn't get the flu shot because maybe they had some kind of immunocompromised disease or they were older. Um, or let's just say they just didn't want to get the flu shot. Shame on you. Well, they're still protected because of herd immunity. So if the rest of the class has been vaccinated, I go in and I sneeze, chances are that virus is not going to make it back to the back row because it's not going to be able to spread and find a connect the dots situation. And that's why after you get a flu shot, sometimes this is when I got a flu shot at the University of Minnesota. I got this lovely sticker that I'm very fond of. It is a public health joke. And if you know anything about herd immunity, you get a little chuckle out of this. So, what does this all have to do with flattening that curve? Flatten the curve. That's all I've been hearing about lately. We got to flatten that curve. Flattening the curve is in reference to this concept of herd immunity. For a novel disease like COVID-19, novel meaning it has never been around before, there is no vaccine against it, and everyone is theoretically susceptible. There's nothing from stopping that virus from spreading around from person to person. There's relatively little or no herd immunity. So there's nothing to stop it from just spreading around. And when you have a disease like COVID 19 that has an R naught above 1, it means that it will spread to more and more people. And when you have a population that is completely susceptible and no one has the vaccine, you're going to have a lot of people getting sick all at once. That is this red curve. And when you have a lot of people getting sick all at once, you have people going to the hospital all at once. When you have a disease like COVID 19 that causes severe respiratory issues, you have a lot of people going to the hospital all at once, and a lot of them wanting things like mechanical ventilators to help them breathe. But we don't have many of those just sitting around. We don't have a healthcare system that puts any value on just having a hospital keep, just an extra 30 mechanical ventilators sitting around just in case. We have just as much as we need year to year, and that's typically fine. When you plop down a new virus where the community has no herd immunity, you get this surge in cases. So, what are we trying to do? We are trying to force herd immunity by all staying home. We are trying to limit the susceptible people that the virus can potentially bump into. So we're almost kind of trying to engineer socially some kind of herd immunity. So the intended consequence of that is to flatten this curve. So think about it's like made out of clay or something, and you just push down on it. It doesn't reduce the amount of disease; it just spreads it out over time. so that hopefully the people that go into the hospital right here can get treated and discharged by the time these people have to go in. And then these people maybe get discharged and treated by the time these people. But if everyone's coming in at once, there's not enough resources to go around. And then decisions have to be made about who gets treatment and who does not. This all has to do with herd immunity. So I hope you enjoyed this little foray into infectious disease epidemiology. Just a few basic principles. Next up will be a more specific discussion of pandemics of zoonotic origin and how do they get here. We're talking spillover, guys. I'll see you soon.